Typically, when cancer drugs are administered to patients during chemotherapy, they can cause a host of adverse side effects, including hair loss and nausea. This is because when these types of drugs are introduced into the bloodstream, they are able to kill not only cancer cells, but healthy cells as well. In order to avoid these adverse side effects, we can design a drug delivery system that specifically targets cancer cells. Our targeted drug delivery system uses a specific characteristic of a breast cancer cell line. These cells overexpress a protein on their surface that acts as a receptor for folic acid, an essential nutrient that is not produced in the human body. This means that on the surface of cancer cells, this protein is expressed at a higher concentration than on the surface of healthy cells. In turn, folic acid is important to cancer cells because of its role in DNA biosynthesis. Increased synthesis of DNA is important to the cancer cells because they are undergoing rapid, uncontrolled cell division. So, how can we use this information to deliver cancer drugs such as methotrexate specifically to cancer cells? Coincidentally, methotrexate is part of a class of drugs called antifolates, which have a similar structure to folic acid. Our targeted drug delivery system attaches an antifolate drug to a polymer, a large molecule composed of repeating units, so that as a whole, the drug delivery system will be able to circulate throughout the body without being excluded through the filtration system of the kidneys. My thesis investigates a specific aggregation mechanism of the folate-binding protein folic acid complex. This aggregation mechanism is important because it can help us understand how the drug distributes throughout the body and near the cells that it will target. We aim to understand the kinetic, thermodynamic, and biochemical properties of this aggregation mechanism using a variety of biophysical techniques. First, we will use force microscopy to analyze the size of these aggregates, mass spectrometry to understand the biochemical interactions between the proteins involved, and calorimetry to understand the kinetic and thermodynamic parameters of this aggregation system. With these methods, we can aim to understand how the folate-binding proteins interact alone in solution, as well as in the presence of antifolate drugs. Studying this system can even illuminate new questions about polymer-protein interactions, as well as how proteins interact at various conditions that are similar to those found in the human body.